Okay, hi everyone, welcome back to another Plant Chats. So as you can tell, today there's a change in the scenery that we're usually used to. So usually when I film these videos, I'm outside in my garden. But today I wanted to film a video indoors because I'm gonna use my microscope today. So let me just put it back like how I usually leave it. So to start this video off, let's talk about the microscope, its functions and its uses. Because today I do plan on looking at some some slides of plants under the microscope and talking to you guys a little bit more about all the individual plant cells and plant tissues and everything. So yeah, first let's start off with the parts of the microscope. So don't mind my microscope, it is a little dirty. I did buy it used, but it's still in working condition. It's still pretty good. Okay, starting from the top, these are the ocular lenses. Ocular as in like eyes, eyepieces. These are the ocular lenses. They have a magnification of 10x. Oh, I guess these are 25 actually. So yeah, they're 25. Down here we have the revolving nose piece. Attached to this nose piece are the objective lenses. Each lens has different magnification. This one, the red one, has a magnification of 4. This one's also called the scanning lens. Then the low power lens has one of 10. And the high power lens has one of 40. This is the oil immersion lens. I've never used it. I have no idea how to, so I don't really care about that one. So right here's the arm. This is where you hold the microscope. When I put it away, I like to hold the arm and the base just so it can have that support and nothing will tip over. And these ocular lenses won't fall out because as you can see, they're loose. One time it did fall out for me and I don't know if it caused any damage, so but yeah, just be careful when you're handling microscopes. So yeah, down here we have the stage. This is a little clip to hold your slide in place. This is the light source. Down here's the iris. It's called the iris diaphragm. Here you can choose how much light is being placed upon the specimen. The specimen is just what you're viewing under the microscope. So this just controls the amount of light. Yeah, and then this also controls the amount of light, but if you like, here you can control how dim you want it or how bright you want it. So right here, these knobs, these are the mechanical stage knobs. So one up here is gonna move it it's gonna move it up and down the stage, and the other one down here is gonna move it left to right or side to side. So these knobs right here and those are the same. So this is the coarse focus knob. As you can see, when I move it, it moves the stage up and down. And this other knob here, this is the fine focus knob. That knob is gonna control how focused the visual is on your specimen. The visual is on your specimen. So I'll show you guys right now when I turn on the microscope. But I just wanted to go over the pieces of a microscope. And well, if you've taken like a biology or life science class, you most definitely learned about how to use a microscope and all of its parts and functions. But yeah, let's get into the content for today. Okay everyone, I'm back. So I have it all set up now. I have it on the low power lens with the light on. We're examining a cotton stem cross section, or the stem of a cotton plant cut horizontally. So this is how it looks under the microscope. I'll go over what we're looking at in a moment. But before that, I just wanted to, let me turn the light off real quick. Before we go over the microscope, I wanted to go over plant tissues. So plant tissues, I have it written down right here on my whiteboard. So first of all, what are tissues? So last time I made a video on cells. So cells are gonna differentiate, do their own little things, have different jobs, and different functions. So when we have tissues, that's gonna be a grouping of cells. So, sorry, I'm like, okay, so for example, let's say we have, sorry, I'm thinking of like specific cell types in the human body. Um, let's say we have, okay, neurons. So a neuron is a cell of like the brain, a nerve cell, nervous system cell. So a neuron. So that's the cell. When we have multiple neurons, that forms neural tissue or nervous tissue. So when you have cells come together that perform the same functions, they're gonna form a tissue. Okay, so we have simple tissues, which is just one type of cell, and we have complex tissues, which is gonna be more than one type of cell. So like two different types of cells working in the same tissues. Okay, so we have tissues covered. Let's talk about histology. So histology is actually the study of these cells and tissues. So pathologists, for example, those are doctors who use histology to examine tissues under the microscope and determine any pathologies that might be present. So like for example, whenever they perform biopsies, they cut a piece off of whatever they want to look at. They stain it and dye it. They look at it under the microscope to observe exactly what, to determine what they're looking at. And well, of course, search for any pathologies. So that's how histology is used in like the medical field, for example. In botany, histology is used to look at plants and look at the different like organs and tissues and cells of plants and determine what their functions are. So we're gonna look at different plant tissues in the stem. The stem is an organ. It's made up of different types of tissues. So yeah, right here I have the different types of plant tissues. So we can separate plant tissues into two different parts, two different sides. So there's permanent tissues and there's meristematic tissues. Whenever you hear meristematic, that's just gonna refer to growth. So meristems in plants are sites where cell division takes place, thus growth will occur in those places. So let's talk about permanent first. So permanent tissues, we can further divide these into ground tissues, vascular tissues, and dermal tissues. So let's go over ground tissues first. So please don't mind my messy writing. I know it's hard to read, but I kind of wrote this in a rush. So we have parenchyma, collenchyma, and sclerenchyma. So they all end the same way in chema, but they all have different functions and they look differently when we observe them under the microscope. So parenchyma, that's gonna be a ground tissue. There's mainly parenchyma in the plants. So parenchyma is like the primary ground tissue. That's gonna be found like literally in everywhere in the plant. So the function of parenchyma cells are to act as storage and to photosynthesize. So this is the primary ground tissue. This is what we're gonna find mainly all over the plant. So yeah, it's mainly just gonna act as storage and photosynthesize. Then we have collenchyma and sclerenchyma. So they're gonna perform pretty similar functions. They will act as support for the plants. So I'm referring to plants that are not woody. By that I mean like not trees or anything. Like these plants, for example, these plants don't have wood, but they need a way to, they need something to help structure themselves, right? So they don't have wood. They're gonna need to move to other parts of themselves to help with the structure, help them stand upright. So yeah, for that we're gonna have collenchyma and sclerenchyma. These have like fibers that help support the plants. The sclerenchyma, sorry, excuse the mess. The sclerenchyma are actually gonna be the strongest out of these three ground tissue. They're gonna have the strongest fibers. Okay, So next, let's talk about vascular tissues. So vascular as in like vessels, like the human, um, like the circulatory system, for example, in humans, made up, composed of the arteries, veins, capillaries, all that. It's the same for plants. So within the vascular tissues, we have two main ones, xylem and phloem. The xylem is going to start at the roots of plants and extend into the stems, leaves, and all that. Xylem is going to help conduct water from the roots to the whole plant. So it's only going to travel from the roots to the shoots being the leaves, stems, all that. Basically, the part above ground. The phloem is going to actually start in both. So it's going to start at the roots and start at the shoots. The phloem is really cool because it gets to conduct both ways. Unlike the xylem can only conduct from the roots to the shoots. The phloem will conduct from the roots to the shoots, as well as the shoots to the roots. Xylem conducts mainly water and minerals. Phloem's going to conduct nutrients, food, food as in like sugars, and the phloem is actually also where viruses pass through in plants. So I've never talked about viruses on this page. Yeah, I don't think so. But I can probably make a video on that sooner or later. But yes. Next, let's talk about dermal tissue. Dermal tissue implants are like these coverings right here, the outer coverings. So dermal tissue is gonna function to protect the plant from the external environment. So we have the epiderm, which is basically just the outermost covering of like the plant, and then the periderm, which periderm we can see in cork bark. So cork bark is this very spongy bark that helps protect the plants that it's growing on. So I don't think every plant has a periderm. Yeah, like these, for example, they don't have periderms. But yeah. Next, let's talk about the meristematic tissue. Apical meristems are going to help increase the length of the plant. So it's gonna grow up and down. So the roots and the shoots. Lateral meristems are gonna help increase the width of the plant. So yeah, not much to say there. I'll probably make a whole video on these soon, because well, I mean that's like the site of growth for all plants. So I'll make a video on that soon, but I just wanted to cover the basics for now. These also include the protoderm, procambium, ground meristem. I'll make a video on this soon. You guys don't need to know this. But okay, now that we've covered basics, now let's go into looking at the microscope. Okay, everyone, I'm back. I've turned the light back on. So now let's look at it under the microscope. So what are we looking at exactly? So this is actually one of the ground tissues that I talked about. This is actually going to be parenchyma. All these white cells, well, clear cells, I should say. So we know it's parenchyma because it has very relatively thin cell walls, as you can see. Thin cell walls. They have irregular shapes. So we know this is going to be parenchyma here, all over. Whenever we have large groupings of parenchyma cells like this is going to be called or referred to as pith. Okay, pith is just large groupings of this parenchyma, these parenchyma cells or ground tissue. Okay, so I moved the slide around and we have here what we call a vascular bundle. So these big tubes right here are actually called vessel elements. Vessel elements and tracheids are going to be related to xylem. And then we have the phloem. The phloem, like I said earlier, conducts food and nutrients. We're gonna have sieve tube elements, sieve plates, relating back to the phloem. I can also see some sclerenchyma and cholenchyma. I'll try to add circles or pictures so you guys can see better. But yeah, this is gonna refer to the vascular bundle. the xylem, the phloem. And I think this might be an open vascular bundle, meaning we could see the cambium in between the phloem and xylem. But again, I'm not 1000% sure. Okay, so here you can better see the sclerenchyma. We know it's sclerenchyma because it has very thick walls. Look at that. So sclerenchyma are actually dead at maturity. So what we're looking at right now is dead cells. Well, I mean, none of these cells are living anymore because they're in a prepared slide, but sclerenchyma are actually dead at maturity. So as you can see right here, the outer edge, that's all gonna be dermal tissue. Dermal tissues, like I said earlier, will help protect the internal environment of the plant from the external environment. Okay, so next we could also see, I believe this should be parenchyma. The parenchyma, when we see parenchyma between the dermal tissue and the vascular bundle, that's going to be referred to as the cortex. Okay, so look at this. Wow, that's really cool, right? So yeah, sclerenchyma, parenchyma, and dermal tissue, the outer edge. So I believe the cotton stem we're looking at should actually, never mind, I won't mention that. I'll probably mention what I was about to say in another video. I don't want to keep this one going for so long, but let me look around in the microscope to see if I can find anything that's worth mentioning. I'll be right back. Okay, I think I should be wrapping up by now. I think I covered whatever I wanted to cover. We talked about the different types of permanent tissue, which was the ground tissue, vascular tissue, and dermal tissue. So we saw examples of each. We saw the epidermis, we saw the xylem and phloem. We also saw some ground tissue like the pith or the parenchyma, the sclerenchyma, and I believe some parenchyma. So that's pretty cool, right? So this is what the stems look like under the microscope. Like I said earlier, the stem is an organ since it has multiple tissue types. Obviously multiple cell types and multiple tissue types, which is, to me, sounds like an organ. So I think that should be all for now. I really hope you guys enjoyed this video. I had a lot of fun making it. But yeah, let me know what interested you and what you learned. So see you guys later. Have a nice day.